Question 1. Which feature best distinguishes eukaryotic cells from prokaryotic cells? A. Membrane-bound organelles. B. Circular chromosomes. C. Cell walls. D. Ribosomes. Correct answer, A. Eukaryotes have internal membrane-bound organelles, including a nucleus. Prokaryotes lack these compartments but still have DNA, ribosomes, and often a cell wall. Question 2. In bacteria, where does most ATP synthesis associated with electron transport occur? A. Inner mitochondrial membrane. B. Infoldings of the plasma membrane. C. Chloroplast thylakoids. D. Lysosomes. Correct answer, B. Prokaryotes lack mitochondria. Components of electron transport are embedded in the plasma membrane, which may be highly folded to increase surface area. Question 3. What is a major advantage of compartmentalization in eukaryotic cells? A. It reduces membrane surface area. B. It isolates incompatible reactions and creates microenvironments, increasing efficiency. C. It prevents diffusion. D. It eliminates the need for enzymes. Correct answer, B. Membrane compartments concentrate substrates, separate pH or redox conditions, and provide surfaces for enzymes, boosting reaction rates and regulation. Question 4. Which organelle is part of the endomembrane system? A. Mitochondrion. B. Chloroplast. C. Golgi apparatus. D. Peroxisome. Correct answer, C. The endomembrane system includes the nuclear envelope, Golgi, lysosomes, and transport vesicles. Mitochondria and chloroplasts are not part of it. Question 5. Which statement about ribosomes is accurate? A. Eukaryotic cytosolic ribosomes are 70S. B. Prokaryotic ribosomes are 80S. C. Ribosomes are membrane-bound organelles. D. Ribosomes translate mRNA. ER-bound ribosomes synthesize secreted and membrane proteins. Correct answer, D. Ribosomes are non-membranous. Free ribosomes make cytosolic proteins. Ribosomes bound to rough ER make secretory, lysosomal, and membrane proteins. Question 6. Which product made in the rough ER is typically sent to the Golgi for processing? A. Steroid hormones. B. Phospholipids only. C. Secreted glycoproteins. D. Nuclear pores. Correct answer, C. Ribosomes on rough ER insert polypeptides into the ER lumen or membrane, where they are glycosylated and packaged into vesicles for the Golgi. Question 7. Which statement about Golgi orientation is correct? A. The cis face points toward the plasma membrane. B. The trans face receives vesicles from the ER. C. The Golgi only degrades proteins. D. The cis face receives ER vesicles. The trans face ships to other destinations. Correct answer, D. Vesicles from the ER fuse with the cis Golgi. Processed cargo leaves the trans Golgi for lysosomes, the plasma membrane, or secretion. Question 8. Lysosomal hydrolases function optimally in which environment? A. Acidic lumen. B. Basic lumen. C. Neutral cytosol. D. High sulf lumen. Correct answer, A. Lysosomes maintain a low pH with proton pumps. The acidity both optimizes enzymes and limits damage if enzymes leak into the cytosol. Question 9. Peroxisomes are best known for which function? A. Protein synthesis. B. Oxidation reactions that generate hydrogen peroxide, then degradation by catalase. C. ATP production via the citric acid cycle. D. Photosynthesis. Correct answer. B. Peroxisomes break down fatty acids and detoxify substrates, produce an H2O2 that is quickly converted to water and oxygen by catalase. Question 10. What is the role of the inner mitochondrial membrane? A. It has little surface area. B. It houses the electron transport chain and ATP synthase on cristae. C. It is continuous with the outer membrane. D. It contains chlorophyll for photosynthesis. Correct answer, B. The highly folded inner membrane increases surface area for oxidative phosphorylation, where a proton gradient powers ATP synthase. Question 11. In chloroplasts, the light reactions occur specifically in the A. Stroma, B. Thylakoid membranes, C. Matrix, D. Inner envelope membrane. Correct answer, B. Photosystems and electron transport components reside in thylakoid membranes. The Calvin cycle runs in the stroma. Question 12. The fluid mosaic model describes membranes as A. Rigid structures where proteins cannot move. B. Static bilayers without cholesterol. C. Dynamic bilayers with laterally mobile lipids and proteins. D. Purely protein structures without lipids. Correct answer, C. Phospholipids and many proteins diffuse laterally. Cholesterol and unsaturated tails modulate fluidity depending on temperature. Question 13. Which best describes simple diffusion across a membrane? A. Requires ATP directly. B. Movement down the concentration gradient without transport proteins. C. Moves solute against its gradient by pumps. D. Vesicle-mediated transport of macromolecules. Correct answer, B. Small nonpolar molecules like O2 and CO2 diffuse through the lipid bilayer down their gradients without protein assistance. Question 14. Facilitated diffusion is characterized by A. Energy input from ATP hydrolysis. B. Use of specific transport proteins and saturation kinetics without direct ATP use. C. Transport of water only. D. Bulk movement via vesicles. Correct answer, B. Carrier or channel proteins move substances down their gradients. Transport shows specificity and can saturate at high substrate concentrations. Question 15. Which is an example of primary active transport? A. Glucose transport by GLUT. B. Water flow through aquaporins. C. Sodium-potassium pump moving Na+ out and K+ in. D. Oxygen diffusion across the membrane. Correct answer, C. The Na plus K plus ATPase uses ATP to move ions against their gradients, maintaining membrane potential and cellular homeostasis. Question 16. Which process specifically uses ligands binding to receptors to internalize substances? A. Phagocytosis. B. Pinocytosis. C. Receptor-mediated endocytosis. D. Exocytosis. Correct answer, C. Receptor-ligand complexes cluster in coated pits and are internalized in coated vesicles, allowing selective uptake at low concentrations. Question 17. An animal cell placed in a hypotonic solution will most likely A. Become turgid, B. Lyse due to water influx, C. Plasmolyze, D. Show no net water movement. Correct answer, B. Water enters the cell by osmosis. Without a rigid cell wall, the membrane can rupture. Plant cells instead become turgid in hypotonic solutions. Question 18. As a cell grows larger, its surface area to volume ratio A. Increases, B. Decreases, C. Stays constant, D. Oscillates unpredictably. Correct answer, B. Volume increases faster than surface area, reducing the SAV ratio and limiting exchange. One reason cells remain small or adopt thin, folded shapes. Question 19. Which adaptation most directly increases surface area for exchange without greatly increasing volume? A. Becoming a larger sphere, B. Developing microvilli, C. Adding a thick secondary wall, D. Filling with a central vacuole only. Correct answer, B. Finger-like microvilli dramatically increase membrane area for nutrient absorption, as seen in intestinal epithelial cells. Question 20. Which evidence best supports the endosymbiotic origin of mitochondria? A. Single membrane and linear DNA, B. Double membrane, circular DNA, 70S ribosomes, and division by binary fission, C. Presence of histones identical to nuclear DNA, D. Presence in prokaryotes only. Correct answer, B. These bacterial-like traits indicate mitochondria descended from engulfed aerobic prokaryotes that became permanent residents. Question 21. In prokaryotes, the primary chromosome is located in the parentheses A. Nucleus, B. Nucleoid region, C. Peroxisome, D. Golgi apparatus. Correct answer, B. Prokaryotes lack a nucleus. Their DNA is concentrated in an unenclosed nucleoid region and may be accompanied by plasmids. Question 22. Which cytoskeletal component forms tracks for vesicle transport and the mitotic spindle? A. Microfilaments, B. Intermediate filaments, C. Microtubules, D. Cellulose microfibrils. Correct answer, C. Microtubules guide vesicles via motor proteins such as kinesin and dynein, and organize chromosome movement during cell division. Question 23. Which junction allows direct passage of ions and small molecules between adjacent animal cells? A. Tight junctions, B. Desmosomes, C. Gap junctions, D. Plasmodesmata. Correct answer, C. Gap junctions form connexon channels connecting cytoplasm to cytoplasm. Plants use plasmodesmata for a similar function. Question 24. Water moves across membranes from regions of A. Lower water potential to higher water potential, B. Higher water potential to lower water potential, C. Lower solute potential to higher solute potential, D. Higher solute concentration to lower solute concentration. Correct answer, B. Solute lowers water potential. Water flows toward the more negative water potential, which often corresponds to higher solute concentration or higher pressure potential elsewhere. Question 25. The proton gradient across the inner mitochondrial membrane powers ATP production by A. Facilitated diffusion through ion channels, B. Secondary active transport of glucose, C. Chemiosmosis through ATP synthase, D. Receptor-mediated endocytosis. Correct answer, C. Protons slow down their electrochemical gradient through ATP synthase, driving phosphorylation of ADP. This coupling is central to cellular respiration.